In an empty surgical suite, time is measured in availability. Right now, over 90,000 people in the United States are waiting for a kidney transplant, relying on the rare occurrence of a matched donor. That massive shortfall is forcing researchers to ask a different question. What if organs were engineered medical devices, manufactured reliably on demand, rather than scarce gifts we just have to wait for? The path to that reality is xenotransplantation, using animal organs to save human lives. But the most intense engineering for this process happens long before a patient ever enters the operating room. The biological factory is a living animal. Modern transplantation has moved past the mechanical challenge of sewing vessels together. The primary hurdle now is brokering a peace treaty between two incompatible immune systems. To build a compatible donor, we drop down past the surgical suite, past the organ, and right into the nucleus of a single pig cell. Researchers locate specific genes responsible for carbohydrate markers, like alpha-gal, which instantly trigger a human immune attack. Using CRISPR, these genetic flags are snipped away entirely. But removing the threat isn't enough. Human transgenes are spliced into those exact gaps, tricking the human body into keeping blood flowing smoothly. In some platforms, scientists take an extra safety step. They use multiplex CRISPR to hunt down and disable dozens of dormant viral sequences, known as endogenous retroviruses, scattered throughout the pig's genome, neutralizing the risk of a viral infection crossing over. This edited single cell contains the complete instructions for the new donor. Through cloning, it is used to develop a living pig with these precise genetic humanizations already baked into its tissue. The resulting animal is a highly engineered organ platform, uniquely calibrated to withstand the hostility of a human immune system. To understand why those genetic edits are required, let's look at what happens when human blood pumps into a completely unedited, wild-type pig organ. Pre-existing human antibodies circulating in the blood spot the pig's carbohydrate flags and lock onto the vessel walls. This triggers a rapid chain reaction of proteins assembling on the sites, punching literal holes through the endothelial lining. The damage spirals rapidly. A severe coagulation mismatch causes human platelets to inappropriately clump and clot, blocking the blood vessels, while immune T cells flood in to dismantle the tissue. Humans and pigs diverged 80 million years ago on the evolutionary tree. That biological gap guarantees an unmodified animal organ will destroy itself within minutes of human reperfusion. Let's rewind this scenario. This time, we'll introduce the genetically edited organ, supported by targeted immunosuppressive drugs, receiving that exact same human blood. Because carbohydrate flags were edited out, the vessel is smooth. Antibodies drift past harmlessly. Human transgenes CD46 and CD55 act as a localized shield, intercepting and disassembling complement proteins before they can cause damage. A specially engineered protein called CD47 acts as a cellular diplomat, delivering a direct "don't eat me" signal to pacify innate immune cells like macrophages. Success in this field requires engineering a biological interface where two species agree on what belongs inside the body, rather than simply suppressing the recipient's immune system until it's defenseless. Zooming out to whole organs, we see a sharp divide in clinical progress. This schematic highlights the difference. The kidney, on the left, operates as a complex structural filter. Gene-edited pig kidneys have functioned in living humans for over 130 days, allowing the technology to enter formal Phase 1 and 2 clinical trials. The liver, on the right, is a vastly more complex biochemical factory. It actively synthesizes incompatible clotting proteins and possesses specific receptors, like ASGSR1, that hunt down and destroy human platelets, leading to massive blood loss. Due to this steep physiological difference, an engineered pig kidney is tested as a permanent replacement, while a pig liver's initial use will likely be as a temporary biological bridge to keep a patient alive until a human organ becomes available. There is a secondary horizon being researched in parallel. What if, instead of engineering an animal's organ to be tolerated by a human, we used the animal purely as a developmental incubator? By knocking out the SAL1 gene, scientists prevent the pig's kidney from forming, leaving a developmental void. Human stem cells are injected into this space. They multiply, filling the exact shape of the missing organ within the pig's framework. To be clear about the timeline, this is early fetal proof of concept science. We are not yet extracting mature, transplant-ready human organs from pigs. It proves the ultimate solution to organ scarcity might eventually diverge from animal organs entirely, moving toward human organs grown purely inside animal hosts. To reach that future, the supply chain has to scale up safely. Donor herds are raised in designated pathogen-free facilities, strictly isolated human-grade clinical manufacturing environments designed to prevent the transfer of animal diseases. The final biological hurdles are steep. Researchers still need to defeat long-term chronic antibody rejection, align highly complex metabolic pathways, and prove these organs can last for decades, not just months. But those months matter. Xenotransplantation has officially crossed the threshold from experimental, compassionate use surgeries into heavily regulated clinical trials. The central question has shifted. We no longer have to ask whether an animal organ can function in a human body at all. The question is whether we can make it safe, durable, and scalable enough to close the waitlist forever.